So I'd like to thank the organizers for inviting me to talk to you today. What I'd like to focus on in the next 15 to 20 minutes is really speaking about the use of repurposed drugs in sarcoma and hopefully shed light on our disease and some potential opportunities with current examples. These are my disclosures. So to start off, to talk a little bit about sarcoma. Sarcoma is a very heterogeneous group of malignancies that arise from the bone and soft tissue of individuals of all ages. The sarcomas are rare. There's estimated to be about 16,000 new cases diagnosed in the United States each year. Actually, I think that's a gross underestimate, but it's kind of the best numbers that we have to work with these days. But to put things into perspective, it's similar in numbers to diseases like testicular cancer, esophageal cancer. And in those diseases, we have very well treatment strategies for, but in sarcoma, we're still defining those. And why is that? Because there's anywhere from 80 to maybe 100 different types of sarcoma out there. So as a community, we really struggle with how to perform comprehensive drug development programs in these rare and heterogeneous cancers. And just to highlight that, this is the 2020 WHO Classification of Sarcoma. And what you can see are all of these different sarcoma subtypes. And to us, these represent different disease entities, right? So when we talk about sarcoma and doing a trial in sarcoma, we wouldn't just do a trial in cancer in general, right? It's how do we begin to understand the nuances of these diseases, the unique aspects of them, and the inter and intra subtype heterogeneity when we think about drug development. And just to take this one step further, this is a list of, say, 30 odd sarcomas that have very specific genetic alterations. And maybe 15 years ago, we would approach a lot of these diseases in the same way. Now, because of a lot of genetic and molecular advancements, we understand, a lot of the drivers in these diseases and are working to understand the disease in a way where we can apply the right therapies for our patients. So when we have a sarcoma clinic of 30 patients, we may be seeing 10 or 15 different diseases, all of whom require, unique approaches in thinking about treatments. And this has really hurt our community. And I think historically, sarcoma was thought not to be a viable field for drug development for many of the reasons that I just spoke about. But the times are really changing. And one of the reasons is the genetic diversity that we're going to talk about is very attractive to drug development. And many of these subtypes are an open field for the application of new technology and scientific advancements. So we're seeing a lot of influx of new agents in trials, and many of them are subtype and disease specific with potential registration tracks. But it puts our community in a difficult situation because we are seeing this swinging tide of research. But as a community, we very much have to figure out how to adapt to this correctly and make sure that we are maximizing our opportunities to study new drugs and technologies in the right disease, and doing so in ways that we're not actually missing signals. And that's an important concept, which hopefully I'll show you some examples for in the next few slides. So how is our community adapting to this area? And I think one of the ways that we're trying to do this is by focusing on subtype specific clinical trials, where we're trying to limit the heterogeneity of the diseases we're treating and focusing on specific opportunities. But this is very difficult for us because when we looked at that WHO classification, many of the diseases or their genomic abnormalities are being newly defined. And as experts, we're trying to understand the context of those genetic abnormalities, these new diseases, in the context of the natural history of the disease. What are the clinical needs of the patient population, and how can we actually improve outcomes? Not only survival outcomes, but also quality of life outcomes in patients with these diseases. And in many of these diseases, bone, soft tissue, we don't have established or validated outcome measures for them. So when we study new drugs, we want to make sure that we're not using incorrect outcome measures, because again, we can really lose potential opportunities. So we have a lot of variables that come together in a sarcoma study, and we want to make sure that we're limiting the variabilities so that we can focus on, the patients and the appropriate drug, and make sure that we're able to interpret the results. And some of the ways that we're looking forward to doing this is actually really focusing on, early development strategies that's really based in biology and science. Again, thinking about single diseases now, single subtypes, and as homogeneous populations as we can, and trying to actually make sure that we're keeping with those homogeneous populations as we move forward with some of the developmental strategies. Very important to understand the technology in the disease and what would be the appropriate outcomes. And a lot of times we have to define new outcomes measures, and focusing on correlative work, biomarkers, patient selections, and really ways to isolate the treatment effect. And this is critical. So with that background in mind, there are three areas that I want to talk about regarding repurposing of drugs in sarcoma. And some of them are very obvious, like the use of imatinib mesylate from CML to GIST, the use of rapamycin in some diseases, and then focusing a little bit more on some drugs that may be simpler to apply, like propranolol in diseases like angiosarcoma, and using these opportunities to kind of highlight, or these examples to highlight opportunities, successes, and challenges. So to start, to talk a little bit about GIST, or gastrointestinal stromal tumors, originally thought to be leiomyosarcomas, or cancers of smooth muscle cells of the GI tract. And GISTs were uniformly fatal when they were locally advanced or metastatic. Here are survivals of 12 to 19 months, respectively. And why? Well, because GISTs were fairly refractory to all conventional therapies used at the time, be it chemotherapy, radiations, or combinations. So there was a lot of need to better understand this disease and to figure out how to move treatments forward. And it was actually some pathological observations that suggested the cells in GIST did not really look like smooth muscle cells, but they looked a little bit more like interstitial cells of Cajal, right? So we now know GIST to be a malignancy of the interstitial cells of Cajal. Importantly, what we knew about the development strata of interstitial cells of Cajal is that they relied on signaling with the KIT receptor from going from a stem cell to a committed progenitor cell, but more so from going from a committed progenitor cell into a mature interstitial cells of Cajal. And so we talk about, the KIT receptor, which is a transmembrane receptor tyrosine kinase. Stem cell factor is the ligand, and upon activation, you can have signaling along, say, the pre-I3 kinase pathway or MAP kinase pathway, which then can allow for transcription and cellular growth. And there was a lot of interest to understand how this receptor was involved in GIST. But there were some genetic diseases known in which patients are born without the wild type receptor, right? And these are diseases such as piebaldism, which was named after the magpie and the bald eagle. And patients have very characteristic phenotypes because of wild type KIT's use in melanogenesis. And this is not something that we saw with our patients in GIST. So it allowed for further discovery. And there were some sentinel papers in the late 1990s. This is by a group from the University of Kyoto that actually showed that it was ligand independent activation of KIT that was causative in GIST because of very specific mutations. So not only did it give us a causative effect in GIST, but it also allowed for some specific immunohistochemistry where we could begin to identify GIST. And almost overnight, the incidence went from about 400 patients to about 5,000 patients. Importantly, it also allowed for the repurposing of a drug. So imatinib mesylate, which was being used to target BCR-ABL in CML, was then applied to GIST. My understanding, and I wasn't involved in the early stories, is that it took some time to convince pharmaceutical companies to actually use it in GIST, but this is the first patient in the New England Journal with very widespread GIST that had a dramatic response. And this led to, now a very famous phase 2 study which showed dramatic responses in patients with advanced and heavily pretreated GIST, and no longer were we seeing some of the survival curves drop off in a year or a year and a half. This led to the approval of, or 2 phase 3 studies that led to the approval of GIST, and subsequently the development of numerous tyrosine kinases targeting KIT in GIST. And now our community is really focused on understanding the correct application of this drug, patterns of response resistance, but opened up a field and really was transformative for patients. Moving to the next example is the use of drugs that target mTOR, and the hallmark of this is rapamycin, which is an old drug. It's produced from the bacterium Streptomyces hygroscopicus, which was actually isolated from bacteria samples from Easter Island or Rapa Nui, which allowed for the name rapamycin. And again, moving into this past diagram, the mTOR pathway is a pathway that a lot of cells use for natural growth, but it's also been implicated in a lot of cancers. So the PI3 kinase mTOR pathway, if dysregulated, you can see cancer growth. Again, a major industry in cancer care, having developed now a lot of rapalogs aside from rapamycin. But this drug was originally used in patients who have had solid organ transplants to prevent rejection, but also shown with the identification of TOR as a potential anti-cancer agent. We're interested in this drug specifically now, and again, having tested it in a lot of different sarcomas and understanding patterns of response and resistance, there is a disease called PEComa, which is a very rare disease. And PEComa is a disease that often has dysregulation of the PI3 kinase mTOR pathway, predominantly with mutations in the tuberous sclerosing complex, which is a negative regulator of TOR. And with this negative regulation of the pathway, you have pathway activation and you get growth of tremendous tumors, often within the abdomen. And this is nice data from Mark Dixon at our group that showed some pretty dramatic responses to patients with PEComas who were treated with rapamycin. And so this was a hallmark of treatment for us, although we fully never really studied it. We never did randomized clinical trials or understood response and resistant patterns because this disease was so rare. Coming now into 2021, Dr. Dixon and Dr. Wagner showed some amazing data with the next generation of a rapamycin, which is an albumin-bound sirolimus. And this albumin-bound sirolimus is now being produced, and there was a beautiful clinical trial, single-arm agent run in PEComa, and what you can see here are dramatic responses, overall response rate of about 40%, median PFS with durable responses of about 11 months. But what was very important with this trial, with a seemingly single disease, a homogeneous population, was understanding the genetics and the response patterns, where the preferential responses to the drug were seen in patients that had specifically mutations in TSC2, not so much, a little variability in TSC1 or if they had PI3 kinase or PTEN mutations. And again, it was predominantly the TSC2 group that were having dramatic responses that were durable, right? So now we are seeing this drug move forward, hopefully in a regulatory process, but the question still remains, is this drug better than rapamycin? And why, as a community, could we not fully develop rapamycin? Does rapamycin follow the same genetic prompts? Would industry ever support a rapamycin study or a head-to-head study? And is this something that the community should support, right? Could we repurpose rapamycin fully in this disease to understand response patterns, whether or not it may be easier to use, better to use, cheaper to use than current strategies, or could they supplement each other, which is critical? Now, just as the last case vignette, I want to move to another rare disease called Angiosarcoma, which is a malignancy of endothelial cells, so blood vessel cells, and we can see it develop in all parts of the body, a very dangerous disease. And we generally classify Angiosarcoma based on where it presents: head and neck, primary breast, associated with radiation. But what was interesting and now known in the community, I'm just highlighting some very beautiful data from Corey Painter at the Broad, who is also one of the founders of Count Me In, was looking at patient samples for people who were actually treated with head and neck angiosarcomas on immunotherapy. And we've been seeing some dramatic responses to immunotherapy in this disease. And what we now know is that head and neck angiosarcomas preferentially have a UVB signature, so maybe high mutational burden, explaining responses, but not everyone with head and neck angiosarcoma responds to immunotherapy. Importantly, there are other subtypes that also respond to immunotherapy. This is data from a clinical trial of Nectar 214, which is a pegylated IL-2 with nivolumab, run by Sandra D'Angelo and Tony Connolly. And what it showed is that there were other groups of angiosarcomas that also responded to immunotherapy: a paravaginal, a bladder radiation associated, an angiosarcoma in the setting of chronic lymphedema, which is Stewart-Treves, and a liver primary angiosarcoma. So significant variability, but our community is a little paralyzed to say, what is the appropriate way to use immunotherapy in angiosarcoma. And what do we need to do to fully understand the correct application, right? Do we need to rechange classification strata? Is this based on location, histology, genomics? And what are our standard approaches? And how can we potentially align patients on the right clinical trials to study this when now many patients are getting immunotherapy off-label in the community, right? So again, brings in the conundrum of how to study this rare disease with a lot of heterogeneity. And why do I say that? Because there's been some really interesting data that vascular tumors, not only angiosarcomas, but another rare disease, epithelioid hemangioendotheliomas, potentially express beta adrenergic receptors. And then if you target these receptors, you can actually see regression in vivo models, cells, and then in vitro models like cell lines, but in vivo models also when the cell lines are implanted into mice and chick embryos. And then there's been some really interesting clinical vignettes. This is a patient with a head and neck angiosarcoma, and you can see how devastating and disfiguring the disease is, that was actually treated with propranolol as a monotherapy for a few weeks, had regression of the disease, and then was actually treated with chemotherapy and radiation and had a dramatic response. And there has been a prospective study of propranolol with vinblastine, and again, seeing dramatic responses. The latter case questions, whether or not the combination is doing anything versus the chemotherapy alone, how to incorporate the heterogeneity of these diseases, and what is the right strategy to use propranolol as either as a single agent or to enhance current therapies in angiosarcoma. But I think it again provides a very reasonable approach for us to go after in sarcoma, to really figure out which patients should get propranolol single agent supplementation with current therapies in angiosarcoma. So I want to end with one of my favorite quotes from James Welch, who's an American poet and writer. And he said, "My poems were just kind of all over the place. They had no focus, no location, nothing. Kind of a series of images that could have been set anywhere. A lot of the poems were just exercises for myself." And in the sarcoma community, I want to be careful that we're just not crossing out poems and saying clinical trials. We really need, especially in sarcoma, to understand, the questions being asked. What is the right population? What are the right tools that we have, especially considering some of the unique diseases we face, along with some of the emerging technologies and drugs. And with that, I'd like to thank you, and I'm really looking forward to discussions today.